Welcome to the cBioPortal tutorial series brought to you by the Genomic Education team at the Jackson Laboratory. This video will demonstrate how to sort study data and specifically how to filter by gene variant on the cBioPortal database. There are times when you are not interested in all the data within a study, but perhaps just a subset of the data. For instance, say you are interested in the data from samples or patients with specific genetic mutations, also called gene variants. It is possible then to filter the data so that only samples with specific mutations will be displayed. To do this, navigate to the Mutated Genes table on the study summary page. Such as that within this pancreatic cancer study, we see the Mutated Genes table on the right. If you are only interested in patients who have KRAS mutations, which is the first gene listed in the Mutated Genes table with a frequency of 91.7%, simply check the box in the third column of the table. Then navigate to the button at the bottom of the table that says Select Samples and press this button to sort the study data. The summary page will automatically reload, and now only the data displayed on the main page will be for samples with KRAS mutations. You can confirm this by looking at the Mutated Genes table, and now the frequency is 100%. You can also look at the top under the study title and see that there's a filter for KRAS mutations. All of the sections of the data are now updated, including the clinical data, all of the charts and graphs on the summary page, only including samples and patients that have KRAS mutations. Let's say, though, that you want to find a gene on the list that is not displayed when you look at the table. The mutated genes list is often very long, including hundreds or even thousands of gene names, so it would take forever to scroll through this list looking for a specific gene. Instead, you can use the search bar at the bottom of the table to look for a specific gene. Let's start by removing all of the filters by clicking Clear All Filters, and then returning to the Mutated Genes table, and say we are interested in the gene PCLO. Typing that into the search bar, we see that the Mutated Genes list filters to only PCLO, and we can again click this box and then hit Select Samples to filter the study data so that only includes samples with PCLO mutations. As we can see, the frequency is now 100%. And now all of the data on the study page includes only those samples with PCLO mutations, including the clinical data. If we click here, we can see that there are only 4 patients included with PCLO mutations. Thank you for your attention. To learn more about the cBioPortal and its features, continue to watch videos in this tutorial series. For other resources from Jack's Genomic Education, find us on our website.